It is so nice to see all you there. It is so.
Hi, David.
It's such a feeling of community. I mean, this image is just beautiful.
This screen. Right. Yeah, just share the screen.
Just go share
the screen. Yeah. And then share, and then 5.
Don't forget to share the sound. To share the sound in a. No, it's not. The sound is not arriving for some reasons.
You have to stop the share and do it again, but there's a little box.
I think correct, as Paul is suggesting. Remember, on the bottom left, you have the sound, and then you have.
Okay. I think we're good. Sorry.
Hi everyone, my name is Marin Scarpelli and I'll be talking about one or almost 2 studies from my PhD. I'm a PhD student here at QUT in Brisbane and I'm investigating how vegetation communities, so how landscape in small spatial scale affect sounds. My supervisors are Dr. Paul Rowe, Dr. Susan Fuller and Dr. David Tucker. I believe everyone here knows that soundscapes are closely related to their landscapes and that landscapes vary across the globe where we have several different biomes. What about small spatial scale? So what about small scale relationships and even more in highly ephemeral landscapes? How does sound vary and what landscape attributes are more important to sounds in a fine spatial scale? So for those who watched Susan's talk yesterday, I went to collect data in the same area. So Barrow is a property 900 kilometers west of Brisbane. It belongs to the Australian Wildlife Conservancy and it's in a semi-arid region here in Queensland. We chose this area for several reasons. It has an abundant bird life because of the presence of a permanent lagoon that provides them with water, which is a very valuable resource in semi-arid regions, but also they have these different vegetation communities in very short distances and the property is relatively flat, so we wouldn't have to deal with topography when analyzing the data. So I placed 24 audio modes in 4 paired transects. So the transects are these 2 lines in here and this other one here. They are in 2 different areas of the property, as you can see from this map. The audio modes recorded for 48 hours and we also measured temperature, humidity, as well as vegetation attributes. Temperature, humidity, and vegetation we measure per point, so each point will have their own measurements, and we did also a bird survey for the whole transect. So we have bird surveys from this transect and from this transect. So here are some examples of the false color spectrograms from this amazing data. The false color spectrograms are the ones generated through acoustic indices, really a combination of acoustic indices. So we have here in red acoustic complexity index, in green we have entropy, and in blue we have events per second. Each spectrogram here is one location, and they are from the same day and the same time. So it is pretty clear how the soundscape is varying across these points. We also have here on the right these photos. They are corresponding to the points where this recording came from. So we can also see how the landscape varies. We have some pretty empty points in terms of the surrounding vegetation. We have some points that are more shrubby, some points with more ground cover, so the landscape is also very variable. Now I'm going to play to you some of the same 5 seconds of sound for each one of these points, so we can also hear the differences. So we're going to start here at point 1. Now 0.2. 0.3. 0.4. And 0.5. So it is pretty clear the difference. I believe we can hear the differences, we can see the differences in false color spectrograms, we can measure and see the differences in landscape, but how do we statistically quantify the differences in sound? So we used acoustic indices. I believe that we are not done with them yet. We know that they are a great tool to summarize big data, and we also know that there are lots of inconsistencies among the results between different studies, between different biomes, between different sampling protocols. But we know, especially from the false color spectrograms, that a combination of indices can tell us lots of information about soundscapes. So, but how do we statistically analyze a combination of acoustic indices? How do we do that? So I came up with this new workflow that uses different methods to analyze acoustic indices. So the input is the wave file, so the recording that came from the environment. We then calculate the acoustic indices and put them into a time series, which is putting the acoustic indices values in order, in time order. So minute 1 will be followed by minute 2, and minute 2 will be followed by minute 3, and so on. And then I performed a motif search, which is running an algorithm that finds repetitive patterns in this time series. So we will find sequence of minutes that repeat along the time series. And I do this for the 3 acoustic indices that we have on the false color spectrogram. So I'll have one time series and one motif search for ACI, one time series for entropy, and one time series for events per second. Then I use these motifs to identify the main sources of sound in the minutes from the recordings. So I go back to the sound, and for example, here we have 4 motifs and the grayscale spectrograms for them. So I know that this first one, it corresponds to a bird. This one, it's an insect motif. This one is a wind motif, and this one is a silent motif. So there is no main sound source in this one. Using these grayscale images and the time series, the motifs, which are these groups of minutes, I then label 30% of the data set, and I feed them into a random forest classification model that will then extrapolate these labels for the whole data set. And we had some pretty good results for this classification model. We had 87% accuracy for biophony, being 77% accurate for birds and 87% accurate for insects, and 88% accuracy for classifying wind, and 91% accuracy to classify silent minutes. Here I have some examples of these motifs. So going back to the spectrograms you saw before, these are the same 5 points, and in white here, this white transparent layer is the motifs for each one of these recordings. We can see clearly that we have some motifs that are related to birds here. We have some motifs that are capturing more the insects. We have some motifs that are silent, and we have some motifs that are predominantly taken by geophony. Okay, but what about the landscape? So I use the motifs to run the landscape analysis. I got 16 vegetation attributes from when I went to the field. They are like shrub cover, temperature, canopy cover, different measures of ground cover. And I used the random forest classification again to select the meaningful features and then run an NMDS and the Permanova. I wanted to split the sound into the vegetation communities they came from based on the vegetation attributes. So when I went to the field, I categorized the points into 5 different vegetation communities, and they go from woodlands to very sparse shrublands. So it's basically a range of shrublands. We had some pretty good results with the NMDS. So we can see here on the left-hand side, we have the shrubby points. So these are the ones that go from shrubland to sparse shrublands. And then here on the right-hand side, we have the very sparse shrublands with the woodlands. The very sparse shrublands and the woodlands are together because they have a low coverage of shrubs in their points in general. And as for the vegetation attributes, we have distance to the nearest shrub and shrub height as the most important attributes to split the motifs in the semi-arid region. We ran the results, the analysis separately for both birds and insects, but we had the same variables and pretty much the same results for both. So we ended up doing all the analysis together. We think that this is because from the bird survey, we know that we have 70% of birds that are either insectivorous or omnivorous, so they feed basically from insects, or they can feed from insects too. And from the Permanova, which is the analysis that tells us how cohesive the groups, the vegetation communities are. So if the differences between groups is bigger than the differences within groups, we had a nice R-squared of 0.49, which is pretty good using just these 2 variables to split the sounds. So the next steps for this are identifying some bird species from some of the motifs. We have started doing this, but I don't have the results yet. It's looking pretty good. We identified half of the birds for half of the points, and it looks like the bird community that the motifs could capture, they are reflecting the vegetation communities they are supposed to be in. And also, after, I want to do some maps to visualize how the shrub cover is spatially distributed, and also how the acoustic indices are spatially distributed. To conclude, we know that soundscape patterns, they vary across vegetation communities in semi-arid regions. We also know that subsequent time series search, so the motif search, is a very good tool to combine acoustic indices, and it can reveal soundscape variation across fine spatial scales, and that in semi-arid regions, shrubs and subcanopy seems to be the main drivers of biophony. I'd like to thank my supervisors, Susan, David, and Paul, both research groups I'm part of, the Australian Wildlife Conservancy for allowing us to go and collect the sounds, and QT and the Australian government for the opportunity of doing the PhD. Thank you.
So, thank you, Marina. And let's see if there are questions. There's a comment from Simon Link.
Jerome.
Yes, please, Jerome.
Hello, thank you very much for this nice talk. Could you please let us know a bit more about the motif search algorithms that you used?
Yeah, I used one algorithm that it's developed to other type of analysis, but it's called HIME, and it basically does this. It searches for the repetitive patterns along a time series. And yeah, I think.
Okay, thank you.
So there's a comment from Simon Link. Hi Nina, what a great first run of results. Although a bit scary. What are the implications for all of us? What are the implications? Sorry. Let's see. What are the implications for all of us, though? If we want to characterize an area, do we need multiple recorders at multiple sites?
Well, I don't have a definitive answer for that, but I see this approach as the same way we have the satellite images. So it depends on the area you're sampling. You need a very fine spatial scale, but if you have a different question, you can sample in a larger resolution. And also, I think it depends on the environment. So you have environment with lots of small vegetation communities. So this is small variation in the spatial scale. And you have other environments, other biomes that are more homogeneous. So I think it will depend on what you're sampling. And also, I think depends on the temporal cover. So maybe if I was sampling in a larger temporal cover, with time, all the points would be pretty much the same. But because I just sampled 48 hours, it was a small snapshot. So the sounds that I recorded, they were more linked to the vegetation, to the small scale, so small vegetation communities.
There's other two questions, actually, even if they are not marked. So from Manuel Vieira, detection systems with very interesting values for birds and insects. Both of these groups encompass many different sound types, species, ?
I didn't have many different sound types for insects, but for birds, there were a high variation. I'm still quantifying the amount of species at this number now, but it was a good sample of what's in the area for 48 hours recording.
There is also another question from Klaus Riede, the Orthoptera expert. Impressive and important data and methodology. Seems you found a method for quick identification of insect sound section. Can you extract them for ID?
If you can identify the insect just from the sound, then it's possible, because we can have both the grayscale spectrograms from the motifs, as I showed, but also we know exactly in the recording which part of the recording the motifs correspond to. So it's really easy to go back and listen to them and see what insect is there if it's possible to identify.
David, you have a sharing Brody.
So, Sharon Brody, oh, thank you.
Hello. Hello. If we have time, yeah. Hello, I'm Sharon in Townsville. How are you going?
Yes, yes.
Thanks for that, Marina. So the idea of motifs is quite new to us, and from what I understand, the motifs are capturing a sequence of like a consistent pattern. So it's picking up the chorus insects quite well and some bird calls. But did I see correctly on the figure? Not the dawn bird chorus so well, because they'd be highly variable over short periods of time, but they're still quite dominant in the soundscape. Yeah, I may be understanding that incorrectly.
No, you're right. It finds the repetitive patterns, but because it's a transformation from sound through the acoustic indices, if the call ends up with the same values for acoustic indices, then you have a repetition. So it's not directly related to the variability of the sound. It's more related to the variability captured by the acoustic indices. But from the results we have, it's different because we are working with 3 different indices. Then what's not captured by one seems to be captured by another one. So it looks like we are getting a good sample of what's going on in the soundscape.
Okay. All right. Thank you.
Thank you very much, Marina. We responded to all the questions that were posed. I think we can go on with the next presentation, which actually from the program, I see it's from yourself as well. Marina, I can see from the program, what does Atlantic forest soundscapes can tell us about the landscape? So
yes.
Again, to you the floor.
Thank you.